net aga zoasi mal gotanu mana lanne sansara sihine So haragiya laso ranghal amavak adure noyenu manalanune
bala velpemi nila kanu peri hantan sihine
Sonduru esanga iye ennam indinu mana sondure antan arane
Shall I start, Takabra?
Yeah.
Good evening, everyone, for joining today's webinar on nanotechnology in COVID-19 control, delivered by Professor Gami Rajapaksa, Senior Professor in Chemistry, University of Peradeniya. So I would like to take this opportunity to apologize from Professor Rajapaksa because in the fly actually we just mentioned a senior lecturer instead of senior professor. So sorry for that, Professor Rajapaksa. So before start the program, I have few announcements. These are very important. So I would humbly request you to mute your microphones during the session. And we have 10 to 15 minutes discussion time after the presentation. And during the discussion time, you can unmute your microphones and ask the questions. If you need to ask any questions or clarification during the session or even after the session, so you can type your question in the chat box. So if you want to ask any question after the session verbally, so you can raise your hand using the sign appeared in your screen. So with this brief announcement, I would like to invite Dr. Vipula Jayampati, the President, Alumni Association of University of Peradeniya, Colombo chapter, to deliver welcome address. Dr. Jayampati, over to you.
Thank you, Mr. Premalal. Dear Professor Rajapaksha, a senior professor and well-known world-recognized professor, and my dear friends who are gathered here virtually from all over the world. Today we are trying to have a very good brainstorming session. For that, actually, I would like to welcome Senior Professor Professor Gamini Rajapaksha, who has invented many things based on nanotechnology. I think everybody can remember, if I show this to you all, this mask with the symbol and this box, right? So that is his one of the most prestigious invention during this COVID period. Dear Professor, we are warmly welcome you on behalf of University of Peradeniya Alumni Association Colombo chapter. And also I would like to welcome all my dear learned friends who are gathered here from all over the world. For some people, maybe good evening, good morning, and good afternoon, because from everywhere, people have gathered here. I know that. So, and also, I would like to welcome all my Colombo chapter of Peradeniya Engineering Faculty members who are behind us every time, and also all my Peradeniya Alumni Association Colombo chapter, all members, and very efficient management council. I would like to welcome again and again for you all. Actually, when we consider about this COVID situation, that's a great disaster all of us, all over the world, we have faced ever. During that period, actually many prestigious scientists came forward to help innocent people like us to protect us from this disaster by introducing many inventions. During that period, I am very proud to say all of us who are gathered here, most of the people who are gathered here, students from Peradeniya University, one of the most beautiful university in the world, and also one of the prestigious university who has ranked under the best 500 among the best 500 universities in the world now, has produced many scientists to come forward and produce many new inventions for this COVID pandemic situation. During that period, Professor Rajapaksha came forward and introduced this nanotechnology masks, first time in the world. Unfortunately, we all couldn't got experience due to this various manufacturing, delivering, and authorization delays. Due to that, we couldn't have this thing. But I think now everybody can experience. So I hope today we are going to have a brainstorming session and very useful session for our lives to protect and prevent from this COVID situation in future too. Thank you, Mr. Premalal. Premalal is one of our forum committee chairman. Actually, he was the former DG of meteorological department. Peradeniya University Alumni Colombo chapter have very few very strong committees. Among that, forum committee is one of the useful committee. They are organizing this such very valuable sessions for you all. In future too, we hope to meet with many sessions. Thank you, Professor. Thank you, everybody.
Okay, thank you, Dr. Jayampati. So the next item is actually I would like to invite Mr. Atula Kuruppo, the former Deputy General Manager of State Pharmaceutical Corporation. Actually, is a batchmate of today's speaker. So I would like to invite Mr. Atulu Kuruppu to introduce Professor Rajapaksa for this session. So Mr. Kuruppu, over to you.
Thank you, Premalal. I am very glad to introduce Professor Gamini Rajapaksa, today's speakers on our special lecture on nano technology. When I am introducing Professor Gamini Rajapaksa, my mind goes back to the 1980s when he entered the University of Peradeniya as a student. He was a very brilliant student in his university career. After the first examination at the university, DSPU, he was qualified to follow the BSc special degree in chemistry. After completing a 4-year period, he was able to obtain the first class honors for the chemistry special degree. Subsequently, he joined the academic staff of the University of Peradeniya, and then he achieved several milestones in his career. He has obtained his PhD from Imperial College of Science, Technology and Medicine in London. Currently, he is working as a senior professor in chemistry department, science faculty, University of Peradeniya. In addition to that, he is a coordinator of the MSc program conducted by the University of Peradeniya in nanoscience and nanotechnology. Professor Rajapaksa is a well-known scientist. He has done his postdoc research in several universities, including Imperial College, University of Bath, University of Central Lancashire, UK, and Max Planck Institute in Germany. He is also a visiting professor of University of Texas and University of Mississippi, USA. And he is regular visiting professor of Shizuoka University, Japan. Professor Gamini Rajapaksa has very broad knowledge and a lot of experience in nanotechnology field. He is a consultant in nanotechnology for clean energy generation in Western Norway University, Norway. Further, he is given his service as a consultant in nanotechnology to several Sri Lankan universities, including Rajarata University, Uvellassa University, and Sabaragamu University. Not only that, he is giving his service as a consultant to the other industries as well. That include Bogala Graphite, TJ, Sarasavi Industries, Isabella and Sarasavi Export, etc. He is also a very good supervisor. He has supervised more than 250 research students. Out of them, he has produced more than 30 PhD degrees. Currently, he has more than 250 index publications. Finally, I would like to mention again, Professor Gamini Rajapaksa is the person who invented the Respiron Nano AV99 antimicrobial phase pump. Now I call upon Professor Gamini Rajapaksa to deliver his presentation. Thank you.
Thank you very much indeed, Atula, for very nice phrasing. And of course, I mean, when my good friend Sarath called me several times to deliver this lecture, I said, actually, I had a lot of other commitments, but yet, I mean, this is the golden opportunity for me to meet our good old friends. And we had a half an hour of a nice chat now. So after about 30, 40 years, even our batchmates, we hardly meet these days. The last 2 years, of course, COVID is prohibiting us to have any personal meetings. But even then, I mean, with these busy schedules, all of us are engaged. So therefore, until we get retired, so it is not that easy to meet. At least I'm very happy that I mean we have met virtually. and I'm very thankful to all of you for giving me this opportunity to sort of brief to you on what we have done and what nanotechnology could do to control this pandemic. So with that very thankful note, I'm going to start my presentation now. I can share. Please enable me to share my screen, please. You have to make me the host.
Yeah, sure. Premalal, can you do that? Yeah, wait, I will do this.
All right. Right. So what I'm going to talk to you today is very friendly manner, the nanotechnology in controlling this pandemic, COVID-19. So the contents go as follows. I'm going to briefly introduce what is COVID-19 and then about SARS-CoV-2 virus, then infection mechanism and lifestyle of SARS-CoV-2 in the host cells. Then I'll show you SCM and SCM, scanning electron microscopic and transmission electron microscopic images of cells infected with SARS-CoV-2. And then we talk about SARS-CoV-2 variants and evidence for reduced antibody protection against SARS-CoV-2 variants. And then we talk about different types of vaccines, risk associated with fast-track vaccine evaluation, as I take from standard journals like Nature, and then ultra-potent COVID-19 vaccine designed via computer is based on some nanotechnology, and nano insights into diagnosis, treatment, and prevention of COVID-19. Then metal and metal oxide nanoparticles in COVID-19 control. And finally, I will show you our R&D activities and what we have done in our capacity to actually to control getting this infection. So what is COVID-19? It is a severe acute respiratory syndrome caused by the coronavirus called SARS-CoV-2. The disease caused by this virus is called COVID-19, as we all know. Since the first description of the coronavirus-related pneumonia outbreak in Wuhan province of China in December 2019, the virus SARS-CoV-2 that causes the infection or the disease, COVID-19, has evolved into a pandemic, as we know now. Globally, as of 15th October 2020, yesterday, there have been 239.4 million confirmed cases of COVID-19, including more than 4.8 million deaths reported to WHO. As of 13 October 2021, a total of nearly 6.5, more than 6.5 billion vaccine doses have been administered. That is where I got it from, covid19.who.in. You can go to that and verify these data. Now you see these things in the graphical mode in different countries, America in this color, and Europe, Southeast Asia, Eastern Mediterranean, Western Pacific, Africa, etc. And you can clearly see there are three, like, we have three maxima in these curves, showing that three stages in this COVID-19 pandemic. That is clearly seen if you take the total cases. You see, in this year, actually, there are three maxima in these COVID-19 waves. So this is the virus. That is this schematic structure of SARS-CoV-2 virus. The viral structure is primarily formed by the structural proteins such as spike S. So you have S1 and S2 spikes. These things are these are the spike proteins, and then membrane M. This is a lipid membrane M, and envelope E. This is the envelope E, and nucleocapsid N proteins. So these are the kind of proteins by which this SARS-CoV-2 virus is made of. The S, M, and E proteins are all embedded in the viral membrane. As you see, all these proteins are embedded in the viral membrane. The N protein interacts with the viral RNA. This is an RNA virus, RNA into a core of the virion. So this is how, say, N protein, the N protein nucleocapsid, this one interacts with that RNA. So this is a virus. The virus is not a live species. It becomes live only when it goes into a host cell. So I'm not going to go into details of this infection mechanism and lifestyle of SARS-CoV-2 in the host cell, but briefly, SARS-CoV-2, then it binds to the spike proteins to your particularly ACE2 receptors in your nose, mouth, etc. They are more expressed in nose, and then it releases a viral genome and ribosome, and then translation of viral polymerase goes into RNA replication and transcription, and eventually to get you infected with the virus. And these are some SCM images. As you see, this is the novel coronavirus SARS-CoV-2. This scanning electron microscopic image, when you zoom this virus is like, their virus is like 125 nanometers. When you magnify it using electron microscopy, you can see them. SARS-CoV-2 round, these are round gold objects, are SARS-CoV-2 viruses. Emerging from the surface of cells cultured in the lab. So you see large number of viruses emerging from the cells cultured in the lab. The virus shown is isolated from a patient in the US. I have taken this from Wikipedia, so it's from a US patient, so it's freely available. I can take them. And basically, when it is infected, the colorized scanning electron micrograph of apoptotic cell, red cell, and in red, heavily infected with SARS-CoV-2. You can see these very small yellow dots are these SARS-CoV-2 viruses. So these cells are heavily infected with virus. So many millions, trillions of viral particles on the cells. And again, and different types of samples infected with SARS-CoV-2. You can see that SARS-CoV-2, it infects in large amounts. And when you can see scanning, you scan the surface of the material. But if you use the transmission mode of the electron microscopy, the electrons go through the sample. You have to have very thin layer. So viruses are very thin. So you can see the structure of the virus. So you see See, this is transmission electron microscope of SARS-CoV-2 viral particles isolated from a patient. You see these particles, so many particles are there. So having understood what SARS-CoV-2 virus is, that is causing this COVID-19, now we know that this virus goes on making different variants. So the original SARS-CoV-2 virus has been subjected to various mutations to result in the evolution of strong and more infective variants. As you know, now Delta, Epsilon variants are more infective and very strong, threatening to the human lives. The currently spreading SARS-CoV-2 variants among the global population include 4 dominant ones, as you know, which are we usually call alpha, beta, like that. For simplicity, alpha variant is formerly called UK variant, or officially referred to as B.1.1.7, B117, first found in London and Kent in UK. And then beta variant, formerly called the South African variant, and officially referred to as B1351. The gamma variant, formerly called Brazil variant, and officially referred to as P1. And the Delta variant, formerly called the Indian variant, and officially referred to as B1617.2. So you see that, we have this real nasty situation because of this Delta variant these days. And luckily we are now controlling the situation, but still, in the first place, our total deaths were limited to 30. Now it's really high because of this highly infective and strong variants coming. Additionally, there are various other variants such as L452R, E484K. Likewise, there are various other variants increasing the danger of current COVID-19 pandemic. You can see various variants. See different UK variant, and then South Africa variant, and Brazil variant, and Indian variant. They are the most prominent and dominant variants of this SARS-CoV-2 virus. See different variants, like antibody-mediated protection after convalescence is high, and this variant reduce. Antibody-mediated protection after vaccination is high for the original virus, and the United UK version is high, and it's reduced there. So likewise, when you get more and more dangerous viruses, the protection becomes less and less. Effectiveness of therapeutic antibodies high for the original version, and then of course high for the UK variant, but reduce and ineffective for South African one, and reduce or ineffective for the Brazilian one as well. See, there are evidence for reduced antibody protection against SARS-CoV-2 variants. Cell culture studies show that mutated viruses are less susceptible to inhibition by antibodies from recovered or vaccinated individuals. Yeah, that can be a person who has got the disease and recovered. From the recovered, then the person has antibodies, or vaccinated people have antibodies. So for both these cohorts, the protection is less for the stronger variants. See, these viruses have mutations in the so-called spike protein. This spike protein, just like these threads, they are the spike proteins, the structure on the surface of the virus that is responsible for the attachment to host cells. So they mutate so that they change these spike proteins to make better catching to the host cells. At the same time, the spike proteins also the major target of the immune response. So in immune response also, the major target is spike protein. So antibodies generated in response to SARS-CoV-2 infection or vaccination bind to spike protein, thereby blocking the virus. The SARS-CoV-2 variants, this one, and are no longer inhibited by an antibody used for COVID-19 therapy. So there are various vaccinations, but these variants are no longer inhibited by. These variants are very strong, so that difficult to be inhibited by the vaccines, as this paper reports. Very high index journal paper. So there are different types of vaccines, of course, as we all know. There are different types of vaccines, and RNA vaccines work by introducing an mRNA sequence which tells cells what to build to the system, which is coded for a specific antigen. So this is RNA. Or DNA, there are vaccines, but not for SARS-CoV-2, but DNA-based vaccines. And viral vector vaccines use live viruses to carry DNA into human cells. Virus-like particles. This type of vaccine contains molecules that mimic the virus, but are not infectious and therefore not a danger. Virus-like particle has been an effective way of creating vaccines against diseases such as HPV, hepatitis, and malaria. Protein subunit. This kind of vaccine uses part of the virus. This is, in this case, the protein component. It uses a protein component. These vaccines can also be used on almost everyone, including people with weakened immune systems and long-term health problems. Then you have inactivate the virus itself. We can inactivate and use it, dead version of the virus. So mRNA vaccine, SARS-CoV-2 virus. So we have spike proteins. mRNA is made with instruction to make viral proteins. So these in the nanoscale, these viral particles are nanoparticles. Nanoparticle is a particle with at least one dimension in the nano range. Nano range is 1 nanometer to 100 nanometer range. So these are actually, most of the viruses are within that nanoscale, but these coronaviruses with crowns are larger than that. Bear virus is like 125 nanometers. Usually these viruses come with aerosol particles coming from your nose or mouth of an infected person. Then the size of this aerosol particle is like 300 nanometers. So usually this spike protein with this virus, we have mRNA is made with instructions to make viral protein. So this mRNA, you give instructions to make viral proteins, is packaged in a lipid nanoparticle. You see, even in this mRNA vaccines, nanoparticles are there, lipid nanoparticles. So these lipid nanoparticles make sure that these virus particles are embedded in these nanoparticles, not destroyed within the systemic circulation, that with the blood, but it goes to right places and do its action, does its action. So these are actually micelles of lipids. So lipid nanoparticles are used to encapsulate this mRNA vaccines. So vaccine is delivered as an injection. So mRNA is released into the host cell. mRNA is used to make viral proteins. So mRNA of the virus is used to make viral proteins inside. So thereby antibodies are developed and give immune response. And if you're interested in more, this is a very good article in CBC News. We can save this one and you can go to this site and read more about it. As we know, because of this pandemic situation, most of these vaccines were approved on emergency or pandemic situation. So classical vaccination phases, like the usual vaccination for other viral infections, like involves preclinical stage of vaccination about 18 to 30 months. And then phase 1 trial, dozens of clinical trials, more than 30 months. Then phase 2, hundreds of clinical trials, more than 32 months. Phase 3, thousands of clinical trials with people, more than 30 months. Phase 4 approval manufacture from 12 to 24 months. If you count, if you add up all this, goes over several years. But in this pandemic situation, you can't follow the standard procedure. So what the approving agencies did was they approved this virus, these vaccinations on emergency basis or pandemic under pandemic situation. So now you see the stages, lengths of these stages have been drastically reduced. Preclinical stage of vaccination, no period. Phase 1, dozens of clinical trials, about 6 months. Phase 2, hundreds of clinical trials, about 6 months. Phase 3, thousands of clinical trials, no period actually. Approval in 6 months or so. So in comparison to the standard procedure that involves several years, more than 5, sometimes 15 years or so, here it is within less than 1 year, everything is done. Therefore, there are some risks associated with this fast track vaccine evaluation, as it is highlighted in this www.nature.com article, Nature article. The administration of a new vaccine must always be carefully associated with a rigorous study of its safety. That is very important. The safety of the vaccine is very, very important. Right? This is particularly important because a vaccine is not a drug for 6 people. So usually when you are sick, we take drugs, but vaccine is not a drug for 6 people. It is given to the healthy people. 6 people at risk of dying, but rather a treatment that is given to those who are well to prevent the risk of falling in death. So vaccine is given to healthy people to prevent the risk of getting that infected. So the race to develop COVID-19 vaccine is not only justified but necessary. But it is, it's important because the number of deaths, as you saw, is in millions. However, the time required to evaluate the dangers and risks that may arise. So, why you need such a long time of phase 1, phase 2, and phase 3 trials is that, you have to evaluate not only the efficacy, but also the side effects, cytotoxicity, what you call dangers and risks that may arise from a new vaccine. So it must be included in its development. In some cases, vaccines prepared against other coronavirus or other viruses have worsened the disease. In some cases, not for this one actually, as this paper tells us, for some other coronaviruses, the vaccines have actually worsened the case. This is induced T helper 2 type immunopathology. Claiming to have won the race to develop COVID-19 vaccine or the distribution of candidate vaccine to clusters of the population before all data from clinical trials are obtained, before obtaining all these clinical trials and carefully analyzing them can be dangerous and erode trust in both the vaccine and regulatory bodies. Actually, this is not a good thing, but it is a must because of the pandemic. But it is associated. It can be associated with various consequences, adverse consequences. See, so then, like recently, people are developing new variants or new types of vaccines also. This is one of those recently developed ultra-potent COVID-19 vaccine designed by computer. Nowadays, actually, drug design. Those days, of course, you remember in our organic research laboratory, people go for these plants, extracts, and then various trials. Nowadays, drug design doesn't follow all these steps. Instead, I mean, you know the disease, you know the mechanism by which drug is interacting with the cell or whatever the molecule. So then they use computational analysis to develop the right thing to block or damage, break or damage the culprit. So based on computational analysis, actually preclinical data published in Cell. Cell is a very high standard journal, show the self-assembling protein nanoparticle. The self-assembling. These self-assembly means these nanoparticles assemble themselves. It is self-assembly is a very old thing actually. All your organs are made of this self-assembly. All your organs are made because no one has made your ear to have this shape or your nose to have this shape. So when the right molecules under right conditions come together, they assemble to give right structures. So likewise, if you know, just like I mean, you are playing with these cards or assembling any other thing, this self-assembly. These molecules under right conditions and right interaction, they assemble themselves in the way to make cells, organs, etc. The nanoparticles does it. So preclinical data published in the Cell show the self-assembling protein nanoparticle vaccine spurs extremely high level of protective antibodies in animal models. So extremely high level of antibodies in animal models. If you have a small amount, it produces large number of antibodies. So now people are trying to develop more and more novel type of vaccines with less and less dangers. And so if you go to this YouTube I will quickly show you how they do
Hello,
it. No,
welcome to the
this is a very
International
good
Conference
keynote
on
speech by
Vitamin
one
D
of our
in Abu Dhabi.
expatriate
In
scientists
this
called
keynote speech,
Professor
I'm going to
Sunil
present to you
Vimalavansa.
on
Yeah,
So
Professor,
this
we
is a
can,
vitamin D
lengthy
and immune
presentation
system,
actually. I
with a
will
we
give
special
cannot
you
see.
emphasis
Sorry,
on
we
COVID.
cannot see. I think you have to share the YouTube
The
screen.
main objective of this presentation
Oh, yeah, we
is
cannot see it.
key
no, I'm
concepts
not going to
related to
actually.
vitamin D,
Sorry.
Okay.
and then we move to the biology of vitamin D.
So I had to share again with YouTube. Yeah.
Of course, you have to share the YouTube, otherwise you cannot see.
Now the thing is that I mean this is a very lengthy presentation. I'm not going to show you the entire presentation. What I'll give you this YouTube address and please go through it. This is like more than 1 hour presentation on how vitamin C is effective in increasing your immunity. So you better go through this YouTube presentation of Professor Vimalavansa. It's more than 1 hour, 2.5 hours or so. And it's a keynote speech done in he's a very good friend of mine living in USA. And Professor Vimalavansa is a distinguished endocrinologist. He knows about these diseases very well. So please go through that YouTube one. But if I am going to show you, the entire presentation will be on that. So since we have no time for that, I'll give you this YouTube link so that you can see it. We talked about, and then we were talking about how can nanotechnology help to combat COVID-19 opportunities and urgent needs. As you can see, the nanotechnology is the technology, very simple technology that manipulates materials in the nanoscale. Nanoscale means at least in one dimension, the size range is from 1 nanometer to 100 nanometers. In other two dimensions, it can be within that range or it can be larger than 100 nanometers. So nanoparticles, nanotechnology can be used to make like auto clean. We can have nanoparticle sprays which will naturally, when you spray onto surfaces, they'll naturally clean the viruses, bacteria, etc. And then as we have done, we can use nanotechnology to make face masks that can destroy SARS-CoV-2 and other bacteria and viruses. And we can make nanotechnically modified clothes with virucidal activity, virus killing activity, so that if the virus incidence on the cloth, it will be destroyed. Then nanotechnology can be used for the detection of virus detection, sensors. And nanotechnologically modified vaccines are there. Nanotechnology based vaccines are there now. People are developing now, still in the developing stage. And even those the standard mRNA vaccines use nanotechnology like lipid nanoparticles to encapsulate this mRNA. And then inhibition of viral infection. Nanotechnology can be used straight away to inhibit the viral infection. And also nanotechnology can be used in many other ways, like improve the immune system response, and also surface decontamination. And so you see in the protection as well as cure, for all these purposes, nanotechnology can be used. So this is one of those strategies using nanotechnology for virus detection and disease diagnosis. So what is shown here is a schematic diagram of COVID-19 FET sensor operation procedure. Graphene as a sensimeter. Graphene, as you know, the single layer of graphite. Discoverers have won the Nobel Prize also. Graphene is the material with highest thermal conductivity and highest electronic conductivity. Graphene is a one atomic layer thick graphite sheet. Graphite is large number of billions of those sheets stuck together just like a carpet. Right, so the graphene as a sensitive material is selected, graphene, and SARS-CoV-2 spike antibody is conjugated onto graphene sheet via this compound. So there is a compound here. The SARS-CoV-2 virus is there. You can attach that onto graphene using this simple chemical material, pyrene butyric acid N-hydroxysuccinimide ester. That is that compound. So this is the linker that can link the virus onto the graphene sheet via this material, which is an interfacing molecule as a probe linker. And then you can detect this virus when viruses comes. So the properties of graphene, when it's attached onto that, when the virus is attached, changes. That change you can measure. So and thereby you can correlate the change to the even number of virus particles, virions. That is one example where nanotechnology can help to detect the virus. And of course, nanotechnology can be used to make personal protection equipment. Not only face masks, all these things that medical personnel who are dealing with patients require these things. So you can use nanoparticles, nanofibers. I show you what we have done towards the end of this presentation. And nano gaps, all these things can be used to make various functions like hydrophobicity, resistance, filtration, breathability, antimicrobial properties. All these properties can be introduced to all these, like the cap, these face masks, face shield, PPT equipment, and then the smart shoes. Even all these things can be modified with nanotechnology to give you maximum protection, particularly suitable for the medical personnel who are dealing constantly with the patients. And then, of course, this is another one, coronavirus COVID-19 nanotechnology. See, you can see, you can virus detection, vaccines, virus control, nano-based personal protection. All these things are there. So, since time is running, actually, I will go into next one. Right. So this is an interesting article where what people have done here is actually binding H cow and this virus to host cell. So using this molecule connected with B boron double AOH2 and this part, you can connect this so and inhibit the virus replication. So nanoparticles, these carbon content dots modified with this material can be used to inhibit the viral replication. That means as a drug can be used. So what nanometers do is nanometers also avoid viral contamination by air, and just like the face mask, and contact with infected surfaces may be especially helpful in sterilizing protective equipment and in a hospital environment. Additionally, nanometers exhibit electrical and optical properties that have been widely explored. So nanometers have special electrical and optical properties, like silver nanoparticles have what is called surface plasmon resonance effect. So those things can be used to make a point of care diagnosis kits. The sensitivity of detection could be improved by using nanoparticles, which enable us to identify the analysis of low concentration. Even low concentration of viruses can be determined using nanoparticles. These strategies contribute to rapid diagnosis and isolation and may facilitate treatment of people infected with SARS-CoV-2. So you see, it can be used to protect as well as to cure. The interactions between nanometers and biological interfaces are the basis for their potential biomedical application. Nanometers interact very nicely with biological interfaces, because biological interfaces are mostly like in the nano range. So they like each other, they interact with each other, and therefore you can use nano technology to not only to detect but also to protect and to cure these viral infective diseases. For example, metal and metal oxide nanoparticles are the very common materials used. I mean, I'm not going to read all these things, just to show you that these particles have very inherent properties. Using them, you can develop various things like inhibition of receptor binding. So these nanoparticles inhibit the spike proteins binding onto your cells, and they can generate reactive oxygen species to destroy these viruses. They can degrade this viral envelope. They can, I mean, I'll show you in our research, they can break down these things and destroy this whole structure. The glycoproteins can be denatured. So in various ways, these nanoparticles can destroy these viruses. Silver nanoparticles are one of those common materials, but the trouble is silver nanoparticles have some cytotoxicity effect to healthy cells. Like we need to use very small concentration, like 1 to 10 ppm, but above that, like concentration about 20 parts per million and above are toxic. So therefore, we have to be careful when using silver nanoparticles. That is why we never use silver nanoparticles in our face mask materials. And we have used actually, we have determined the antiviral activity. This is our research now. Titanium dioxide, copper oxide, and zinc oxide nanoparticles against respiratory syncytial virus, RSV. This is, I mean, in Sri Lanka, we have to have actually very third level 3 safety cabinets, safety rooms to work with SARS-CoV-2, but in collaboration with Professor Fasiha Noordin. She is the virologist in our microbiology department of our medical faculty. We evaluated the antiviral activity against this similar virus called respiratory syncytial virus, RSV. So we see that titration and dioxide nanoparticle concentration percentage 0.5. Without that, and RSV antigen cells per field, 30 survived out of 50. So as you increase the concentration to like 0.1 to 1 survive. Again, of course, the results are not actually proportional. You see, as you increase the concentration, the number is again increasing. So we use this copper oxide. She has given copper. Copper oxide also, we see with even very small concentration, 0.06, like 2 out of 50 survived. 0.1 to 1 out of 50 survived. So with very small concentration, you can kill the virus or destroy the virus, but trouble is copper oxide is also toxic. Although in America also now copper oxide based face masks are available, copper oxide is not very friendly for us. So although we have determined, we never use copper oxide also. And zinc oxide is the material that we have used. The nanoparticles, as you can see, without zinc oxide, with time also, the virus destroys, so 30 out of 50 remains. But within that time, if we use we use 3%, but if we use half the dosage, 1.5%, nothing remains. Even the quarter dosage, nothing remains. 0.37, that means 1/8 the dosage, only 1 out of 50 remains. So fields, not only the virus fields we are talking about, large number of viruses in one field. So you see that zinc oxide nanoparticles are highly efficient, very efficient to destroy this RSV virus. So those days, because we did not have facilities to do this actually SARS-CoV-2 test, we worked with bacteria and this RSV virus. But then, of course, this test can be done from London Virological Research Services, which is a part of Imperial College. And because I'm an alumnus of Imperial College, they agreed to give me at a lower rate, like it's still 5,500 pounds a test. That is like 1.7 million rupees. So nevertheless, we did it. Otherwise, it's 13,000 rupees, 13,000 pounds actually. So we did that with SARS-CoV-2 also. So this is how these tests were done. I think I don't want to bore you with all these procedures, but this is how our experts in virology did the RSV test using fluorescence microscopy visualization. And they confirm that dose-dependent activity is obvious with zinc oxide only, not with others. So we use zinc oxide nanoparticles as viricidal materials. But the nanoparticles that we use were not spherical, but the core is spherical, just like the virus. We have this star shape. The sharp blades coming out of this sphere in the middle. So in all directions, these blades in the nanoscale are very, very sharp, so that they can basically damage the spike proteins, cut the envelope, and destroy that virus into pieces, as I will show you later. So that is the major action. So most of these the microbes can develop resistance to chemical agents. However, for this mechanical action, just like you're cutting the parts and bits and pieces of the virus using scissors, these sharp blades of nanospheres can destroy and break and cut into pieces. So that is how they destroy virus and bacteria. So we find that zinc oxide is very effective in doing that. So we did with various bacteria. I mean, actually, we have done these experiments for several years. I have produced 3 or 4 PhDs in like zinc oxide based textiles, antimicrobial textiles, starting from Dr. Sri Mala Pereira. She is the head of Department of Textile Technology Department of University of Moratu, actually. So we see large inhibition zones with this bacteria do not grow. Even if we, that is because, I mean, these nanoparticle surfaces are positively charged, viral envelope or bacterial envelope is usually negatively charged. So therefore, the electrostatically they attract not only on the surface but also in the vicinity. So that attraction, what we call, I mean, that is called the Nernst diffusion layer, has a limit. So within that range, the bacteria cannot survive because it comes to the cloth and modified cloth and destroys. So with 3% zinc oxide, so they have tried 3% zinc oxide with 1% copper oxide, etc. With control. Control, there is no that. So with cloth 3 zinc oxide 3%, small, I mean, doesn't bacteria on the cloth all dead, and also in the vicinity also a small circle bacteria. When you add more, at least 1% copper oxide, the cumulative effect, synergistic effect makes it even more strongly attracting, so that the inhibition zone becomes larger. So tried with 3 different, like gram positive, gram negative, and also very dangerous bacteria like Pseudomonas. And then actually these our mass we got tested from Bureau Veritas. Actually we have to do 13 tests. Bacterial filtration efficiency. When you tested that with, actually, if you do that particle filtration Syntec, you don't get the accurate values, because it in Syntec machine, what they do is not correct actually. The particles in the air is sucked through the mask and then analyzed. But it depends on how much particles in the air. So usually polluted air should have at least 10 to the 20 particles. So it is not a good thing, but it also measures only the neutral particles. But since bacteria viruses are charged particles, so you have to use bacterial filtration efficiency. So which we did from ITI, and we got 99.99% bacteria filtration through our modified cloth. I'll show you. Then splash resistance. So at a particular pressure, you splash blood onto the mask material or the mask and then measure whether blood penetrates through it. So that is important for surgeons. So the rate is like higher rate than the blood coming from the cut arteries. And then also differential pressure. That is when you have a mask, there is a pressure difference. Pressure is created inside. So without mask, there is no pressure difference. So the pressure difference between your face and the mask in the head space is called the differential pressure, which is measured for a good breathability. This differential pressure should be very, very low. Imagine it's 4 water millimeter per square centimeter. So our mask pass that also. And then dimension of the mask. So with certain dimensions are there, it passes. Dimension on the nose piece also dimension pass. Then number of pleats and layers in the mask pass. Safety, is a microbiological cleanliness pass. Flammability. So this, because our masks are made of cotton materials, unlike this polypropylene materials, so the cotton takes time to ignite. So usually if you are wearing a mask and if there's sudden fire is there, so we need to give time to remove the mask. So at least 10 seconds. So usually these, I don't want to mention names, but polypropylene type masks that I use have like less than 10, this is 10 seconds, that minimum one. However, this cotton is not that flammable, so therefore it takes about 20, 30 or even 1 minute to burn. So that means we are giving enough time. So flammability is best passed with this cotton mask, and pH and all these parameters pass. And also there are ample literature in ACS Omega and various other standard journals on how zinc oxide nanoparticles can destroy SARS-CoV-2. And then, of course, our masks are designed to make them usable for repeated washing. One mask can be used at least 25 times washable in standard washing machines. So within we calculate zinc oxide ppm hardly changes. You see 10,599, almost 10,000 similar range. Even if you wash for that is because these nanoparticles are chemically bound. So that we did this research on particularly on mass materials for more than 1 year to make sure that they don't detach. And then we pass an airflow at a rate much higher than your breathing rate. Breathing rate is 6 liters per minute, but we pass airflow at 10 liters per minute or higher for 24 continuous hours and measured both the titanium dioxide microparticle and zinc oxide nanoparticle amounts. There's no change whatsoever. And then percentage reduction of bacterial count against control sample 0, but this 99.89, 99.32. So almost higher than 99 point bacterial count reduction, even after 25 times wash samples. Wow, so many people are coming in the waiting room now. And also the safety. So that was an issue that the microbiologists brought for several more than several months to get a NMR certificate. I mean, we know that they are non-toxic, but then we had to do this toxicity and hypersensitivity test using mouse models, and we had to sacrifice like 50 mice, unfortunately, but not me. The Professor Jayant Rajapaksa, professor of veterinary pathobiology in our vet faculty did it. Our product name Residon Nano EV99 face mask, and over 3 months. So then what he did was he had a control mice sample and then the test mice sample. For the control mice sample, the normal air comes. For the test mice samples, air always comes through the mask. They don't get any normal air coming from the mask. It's actually normal air, but it's coming through the mask. And then just before treatment and 3 days after, 15 days after, 30 days after, 6 days and 9, over 3 months measured the normal air. There's hardly any change. This is the weights of the air through mask, the mice subject to air through mask. The weight doesn't change. And also measured the histopathology of lungs, kidneys, livers of normal air exposure and air exposure through mask after 3 days, 15 days, etc. So then you see that there's no control 3 days, up to like 3 months. That is for mice, 3 months is more than enough. So there's no change whatsoever. So that means that is because we knew that because our nanoparticles, our materials used in these fabrics are chemically bound and are not detached due to airflow. So there's no chance for you to inhale them. So we have done this test for 3 months, and we have this big report, part of that I have put there. And the conclusion, there are no any pathological lesions observed, either hypersensitivity or deposition of collagen, and these are the things that can deposit if a particle goes. Proteoglycans remolding the extracellular membrane of underlying epithelium of the air passage. Therefore, there is no any toxicity effect to lungs, liver, and kidney tissues. Based on the above study, the breathing through Respiron Nano EV99 face mask has no detectable adverse effect in acute or chronic exposure in the respiratory tract or the lungs, I don't know what is cyanomatous tissues. Dr. Rajapaksa is a chair and senior professor of parasitology. So then it's great. So then actually these microbiologists were against this one, but then they could not go for that because we have the report saying that it is not toxic at all. We knew that, we have proven that, but they saying that particles do not detach, but they demanded this. Actually, we did it. And also, I mean, we can measure titanium dioxide and zinc oxide contents using XRD before wash, after several 25 washes. You see the amounts measured using XRD hardly changes. See, hardly changes. So this is what I said before air passing, after air passing for at high rate. For 24 hours, there is hardly any change. So 45.6, 19.2, 45, 20, almost the same ratio. That shows. So this is a video that you can enjoy about our face mask, and I'll brief you on what they are.
Respiron AV99 is an environmental friendly, biodegradable, low cost, reusable and antimicrobial face mask with water and blood repellency and stain resistance. It also repels and destroys any aerosolized viruses or bacteria. In this multifunctional face mask, the target has been to reduce both the waste burden and the impact on the environment. As such, cotton, a commonly available, natural degradable fabric material, has been used in 3 layers, nanotechnologically modified individually, to have unique, inherent properties. The outer layer cotton fabric is super hydrophobic to repel any aerosolized viruses and microbes, as well as blood or any other water-based stains, comparable to a lotus leaf shedding water. Any microbes penetrating the outer layer will reach the nanotechnologically modified middle layer with multimodal antimicrobial activity. Micro and nanoparticles are used to partially block the pores of the woven cotton fabric, while chemical bonding to a level of 300 nanometers, resulting in nanometric range filtration. The adjustable ear loops ensure tight fitting to the face of the wearer. A unique mask design and a metal or plastic nose plate addresses the problem of air escape at the nasal bridge. The innermost layer is made up of 100% hydrophilic cotton to absorb carbon dioxide and moisture present in the exhaled air. This face mask can be reused even after 25 cycles of washing or disinfection, at least for a period of 1 month. The nanoparticles used in these masks are safe in a wide range of applications, including medical and cosmetic industry. High airflow tests and wash cycles have proven that the chemically bonded nanoparticles are stable and do not detach from the fabric surfaces. The mask has medical-grade characteristics with high resistance to flammability, satisfactory results in differential pressure tests for breathability, and is highly resistant to blood and fluid permeation.
So this is the team, myself, Dr. Chamindera, subject radiologist, and my So I will quickly go through this is the outer layer. You see that if your blood comes, it stays as a sphere. When you tilt it, it falls off, just like the water on the lotus leaf. So this is with the superhydrophobicity. The dislikeness for water is measured by measuring the contact angle. So if the contact angle is above 150, we call the material is superhydrophobic. If it is above 90, a contact angle, it's still hydrophobic. Below 90, it likes water, hydrophilic. So you see at least up to 20 you see is super hydrophobic, and up to 25 even 30 is hydrophobic. So it's so therefore we recommend, because this is the thing, because due to wear and tear old days, it's possible to physically damage. If physical damages when you wash so many times, you see contact angle remains above 150 for 20 washing cycles, and even 30 washing cycles you can go still hydrophobic. So after that we don't recommend, because this property gradually decreases. But other properties down, so at least 25 washing cycles you can go. So here that's how we, cotton, when you, it's a woven fabric, we have yarns, say horizontal and vertical. Between them you have pores. So we use a very high sensitive light microscope, and you see that the cotton surface has pores. As you block these pores, gets reduced. As you block completely, the light cannot go through at all. So there are that means that pores are reduced down to at least the smallest number of visible wavelength, that is 400 nanometers light. Actually, when you do at SCA, you see that you have reduced down to 300 nanometers. That we did by blocking with right size. These are like 30 micrometer pores, so micro particles of titanium dioxide, the surface of which is positively charged, so that if virus comes to mid layer, then it is electrostatically bound and destroyed. And also these pores do not let any aerosolized particles to mechanically go through also. So you see that with different extent of coverage, you see different extents at 0.3 micrometer, 300 nanometer is the size of the aerosol particle. So when you measure from sling tech, still you get 67 plus 4 with that. So as I said, sling tech measurements are not reliable. Above that you get 91%, but this even if you do N95, you don't get 95 is sling tech, because the method itself is strong. But when you do bacteria filtration, we see that with that we get 99.99% bacteria filtration efficiency. And this is RSV antigen test results. These are bacteria results. Even with Pseudomonas, is a very dangerous bacteria. With 3%, you can kill all of those bacteria. This is ITI test report. Bacterial filtration of sample 99.8 knot, sample 2 99.99, 99.99, 8 knot, 99 like that. So 8 knot to 99.99% bacteria will make sure that these size particles do not go through this middle layer. And the inner layer is a soft inner layer with excellent moisture absorption. So actually, this is demonstrated like this. You put some dye, it immediately absorbs into that. So in the perspiration and in the exhaled air, you have carbon dioxide. So you want to get rid of that carbon dioxide. You should not let carbon dioxide accumulate between your face and the mask. It's the head space. In some masks, of course, this carbon dioxide accumulation is there. That is why you have one-way valve. So the one-way valve in those masks, but that is banned because if you are a patient, then of course, I mean, all the viruses, the aerosol particles immediately go into the air with the one-way valve that is open out to the atmosphere, towards the atmosphere. So that is banned. In our case, it is quickly absorbed because cotton, it likes water, it absorbs and quickly removes from the headspace. So there's no, because I'm actually jogging for 1 hour, like 1.2 kilometer path, 6 rounds at least, and with wearing this mask. I don't get any problem because the breathability is adjusted to the right level, and also this carbon dioxide and water escape is also there because it's cotton. So inner layer with excellent moisture absorption, wicking and evaporation, free of fluid clogging, and non-accumulation of carbon dioxide and no re-inhalation. Some of these masks which have poor carbon dioxide absorption gives the carbon dioxide rich air in front of your face, in the headspace between mask and the face. And then because carbon dioxide is heavier, when you inhale, what you are inhaling is always carbon dioxide rich air, so oxygen concentration not enough. There were cases where the mass wearers were admitted to ICU even. So you have to be careful. So the material like these things, we have taken all these things to make sure that you have no problem of carbon dioxide accumulation and reinhalation. It's a barrier between phase airway and filtration layer. So therefore, even if they are not the material that used to modify in the inner and the outer layers, so this is a barrier. So whatever the material, they are the materials that are used in face creams also, but we make sure that they are not even touching your face. There's a barrier. It's a barrier. Unmodified super hydrophilic cotton barrier. So comfortable to face because it's cotton, safe and non-allergenic. So we have tested with 500 people, randomly selected, and there were no complaints of allergic reactions or anything. So we have given a questionnaire with several questions to answer yes or no. So with 500 people, and its ease of wearing, all these properties are there. And actually, if the time permits, I can quickly show you, but time is not permitted now, almost 8 o'clock. Those days, there were no instruments, so we had to build the instruments. So I'll quickly show you a couple of instruments and wind up this one. This blood penetration tester. This is one that our students developed. So only a couple of minutes, so I hope you can bear. So this actual blood is splashed on the mask at a rate of blood coming through cut arteries. So you put a mask there and splash blood onto it. These are the masks that we in the development stage. Before perfecting the shape, etc. So blood is splashed on the superhydrophobic outer layer. When you are wearing, it comes to that one. You see blood clot, blood spear like that, but the other side, there's no blood. The next one. Oh, I see. Oh, yeah, this one. Sorry, the previous one. So this is a differential pressure tester, all computerized, automated delta P equipment. So air comes at the rate of breathing, and this is stainless steel disc. We have 2 discs of stainless steel. In between, there are holes in the middle, mass is sandwiched in between, and then before without mass there's no pressure. With mass we measure pressure difference, so it comes below 4 water millimeter per square centimeter. And flammable test also we developed. It measures the time taken for the mass to burn out. when it's admitting, it stops. You ignite and then automatic watch measures the time. Nowadays, you can measure time up to 3 decimal points or seconds. So it's well past the limit of 10 seconds because it's cotton, and modified with inorganic particles means even takes longer time. That's 28 seconds. This one. Sometimes actually these are experimental stage. Now we have improved that even further to make sure that even in a fire and you are safe, you can have enough time to sort of remove your mask without catching fire. So safety, no change of micro and nanoparticles after 2 intervention cycles. Chemical attached to fabric, no direct body contact of modified cotton, no irritation or other adverse health effects, no untoxic as per mouse model test, commonly used in body and face creams and lotions. This environmental friendly material, that is also very important because most of these polypropylene type of mask, imagine there are like 7.5 billion people in the world. So all of them are supposed to wear these masks. So these like polypropylene masks are meant to wear only one time. So therefore, every day you have to throw 7.5 billion plastic pieces. So that will have enormous effect on the environment, and you can't imagine the kind of diseases that you may expect due to those things. The plastic pollution. So to overcome these problems, we have used cotton, and then we have modified in different way to have 3 layers to have different effects, so that this 100% sure face mask that sort of that prevents the virus getting into you, or if you are a patient, the virus particles have to go through the middle layer, and then when they come out, they are destroyed. So you don't get the virus, you don't give the virus either. So there are some references also. Finally, what we are trying to make now is, this is air has enough oxygen. So, but if you selectively remove nitrogen and carbon dioxide in air, you get pure oxygen. So the oxygen concentrate usually have like 93 to 94% pure oxygen, but with our one we can get up to 96.7, near 97% pure. That is this is from air, so you don't need oxygen tanks. So air has 21% oxygen. So what you do is you use a zeolite to absorb nitrogen and carbon dioxide. Before that, we remove moisture and get pure oxygen. So we have developed this one actually, and it's all automated. We have 2 cylinders. One every time this flow is, and every 6 second flow is directed to one. So this is pressurized to absorb nitrogen and carbon dioxide. Meantime, this is the absorbed one is released. So every time shifts into 2. So continuously you can operate this one. So we are building that one, but to get oxygen from air, pure oxygen from air. So that's all that I want to discuss with you. It took a lot of time because of these technical problems and everything. I hope you can bear with me and you enjoyed it. And thank you very much. If you have any questions, I'm very pleased to sort of tackle your questions. Thank you.
Yeah, thank you very much, Professor Rajapaksa, for delivering informative presentation on COVID and its evolution and different variants and even vaccines. So even though nanotechnology has a long history, as mentioned, many people heard about it with this pandemic situation. I think Professor Rajapaksa explained many things related to COVID-19 and also nanotechnology and its application, research he has done on nanotechnology. So I think so we are running out of time, so we have to stop at 8:30. So there are a few questions in the chat box. I would like to ask those questions from Professor Rajapaksa. I think you better give that very quick answer for these questions. So one question asked by Mr. Iftas. So what are the available tests for newly invented masks that we can conduct to ensure to wear? I think you already explained it. So do you have anything to?
Yeah, actually, I mean, not all mask. This is the only mask that is antiviral in this country. So therefore, we have to make sure that it kills SARS-CoV-2 because it's meant for SARS-CoV-2 and also other viruses and bacteria. So these tests we have done. Safety tests, biocompatibility and toxicity tests we have done. And also all the other tests like 13 tests I told you, the breathability, and flammability, sizes, bacteria filtration, particle filtration, and because our one is superhydrophobic outer layer, so they are superhydrophobic property, all these things, and splash resistance, blood penetration resistance. So these are things that we have to measure for our mask. But normal mask, say if I tell you N95, it has like at least 95% filtration at 300 nanometer only. It doesn't have any antiviral or antibacterial effect. So therefore, it doesn't require all these antiviral tests. But in our case, we did antiviral tests against SARS-CoV-2. Actually, that slide should be there. At Imperial College, this London Virological Research Services, and we in the past it confirms that it kills SARS-CoV-2. And actually, every batch we are monitoring this we are giving samples to BV to measure all these proper 13 tests that they are supposed to do. And then, of course, because our one is environmental friendly material and reusable, and we have to measure all the properties after washing also. So each batch we give samples to Bureau Veritas because we can do it, but our reports are not accepted. So we have to get a standard test done from reputed accredited institutes like Bureau Veritas or ITI. So we get all these tests done for each batch, samples from each batch. But for normal mask, actually, if say a surgical mask has only 15 to 30% filtration at 300 nanometer, that is aerosol particles. But bear virus can go through it, but only aerosol particles, there is 15 to 30% filtration. That is all that it has, and it is one-time use, so it doesn't guarantee that you don't get it. Even the N95 has problems because, as I said, this carbon dioxide accumulation in the headspace is a problem. So all these things we have to measure and make sure safety and whatever the property that you claim should be proven by not by yourselves. Actually, you have to do it yourself first, and then from a standard tests from accredited institutes.
Okay, thank you. Thank you, Professor Rajapaksa. So there are a few more questions. I think so. We don't have much time, so we have to stop.
We can quickly go.
Yeah. So the second question is from Ms. Shriyani. If someone has already got COVID disease and GERD, is it necessary to get vaccinated?
Well, the person who has got COVID may have antibodies developed, but still, I think it's safe to, I can't talk much about vaccination because I'm not a medical doctor, there are various research articles coming against vaccination also. So therefore, I don't like to make comments on those things. It is not my field.
Damini? I think this week there was a publication. Once you get the COVID, you are 15 times better than the vaccinated person.
Exactly. Yeah. Yeah. If you survive from the COVID, I mean, these antibodies that you develop are actually against that, isn't it? Unlike these dead viruses, isn't it?
Yeah, they say it's 15 times. It's
15
quite high.
Yeah.
Quite high.
So,
Thank you.
Okay, thank you very much. So the third question, so Mr. Nuwan Kumar Singh, despite the numerous advantages of using nanotechnology in many fields, including COVID applications, is there any mechanism to control the possible environmental contamination by using this new technology? I think yes.
Well, I mean, environmental contamination depends on the material. Like, I mean, if nanoparticles go into atmosphere all the time, constantly, like in the workers of mines, etc., they may get troubles. But there are research articles showing that the zinc oxide miners have a higher longitude than normal workers. So it depends on the dose, actually. But I mean, when you use these things, you have to make sure that they are not sort of they are not going to the atmosphere. In our case, we have made sure that. But then these nanoparticles are natural materials. They are present on the Earth crust anyway. So therefore, once you throw our mass, actually they undergo biodegradation. So therefore, there is no harm. But only harm comes when too much of particles are inhaled. If you inhale too much of particles, of course, you can get trouble. So we have to make sure that they are not inhaled. In a way, when you are wearing the mask, these days, you don't even get common cold. So that is, you are protected from many, and wheezing also, protected from getting nanoparticles into your lungs.
Okay, the third one, the fourth one, what is the Mr. Sarath asked, actually, what is your opinion about using the medicine called ivermectin to treat COVID-19 infected patients?
As far as I know, none of those things are actually 100% like made for COVID-19, but when the pneumonia is a kind of drug that is given for the chronic strong pneumonia, acute pneumonia situation. So if you, I mean, the people die because of the pneumonia. So if you can treat for that, then people will not die. Patient will not die. So in a way, it's good, but it is given under strict control of doctors and under strict control of situation. I mean, a non-medical person like me cannot comment on that actually.
Yeah, Professor Rajpaksa, can you stop your screen sharing and stop?
Am I sharing the screen?
Yeah. So then we can.
Stop share. Yeah. Sorry about that. Yeah. Okay.
Yeah. And can you exit from the host? Is possible?
How do I do this?
And make me host.
Okay.
Yeah.
Where is that host thing? Is it record reactions host live on Facebook?
Click on participants and then click on
participant. All right. Okay.
Participants, and you select me, Priyat Kabral. Select me,
and then make him host. Select Kabral and make him host.
I clicked on participants, but I don't get.
Then click again. Click again one more time.
Invite only. Manage participants.
Now click on the person. Click on the person right hand side. Then you will get you can make it host.
Which one? Participants?
Yeah, under participants. Under participant.
I get only invite now.
When I get a list of people, no participants.
Yeah.
You get a list of participants. Then only you have to make him host.
Where is it? I'm not getting.
No, when you click on the participants, you get a list on the right hand side.
Yeah. It is
separate list.
Okay.
There you have to select Cabral.
Yeah.
Cabral. And then?
And there is this thing called
Mo.
Yeah.
Chat to stop video pin make host. Yeah, make. Oh dear, I did it.
Yeah.
Thank you.
So
you can do nanotechnically, but you can't do the simple technique.
Yeah. So actually, we have only 9 minutes. We have some more function. That some few function. So I'll ask one more. So you better give a quick answer. Respiron mass test have passed the bacterial filtration test. How about failing the viral filtration test since COVID-19 is a virus?
Actually, COVID-19 is a virus, so we have done that test at the Imperial College. Actually, didn't I show you? Actually, I had a test report saying that it kills the COVID-19 SARS-CoV-2 virus. That is what I said. It costs like 1.7 million rupees. So the company making this spend that much and send a sample. They are going to send another sample now with the new batch also. So the SARS-CoV-2 test passed. And also RSC test pass. RSC 100%.
Okay. So the Pubudu Vimalasiri, can you go into a little bit more detail as to how the zinc oxide nanoparticles are chemical? I think so.
No time, I think, to go for details.
No time. Yeah. So you can go.
We can literally catch about that. Yeah.
So you know him. What is the efficiency of the mass in destroying COVID-19 viral particles?
It's 9 point.
Just okay.
Yeah, something comes, but we are going to send again.
Okay. Okay. So I think so. There are some more questions. So there are no time to ask all those questions. So sorry for that. And the final item is hand over the memento. So I would like to invite. So this is to hand over the memento to Professor Rajpath as a token of appreciation. I would like to invite Dr. Vipula Jayampathy to hand over this memento to Professor Rajapaksa. First, I would like to show this one. You can see. You can see this in our screen.
Yeah.
Can you see it?
Yeah.
So presented to Professor R.M.G. Rajapaksa by the Alumni Association of University of Peradeniya Colombo chapter in sincere appreciation of contribution as an invited speaker of the webinar on nanotechnology in COVID-19 control on today. So I'd like to invite Professor Dr. Vipula Jayampathy. Yeah, Jayampathy, can you say about something?
Yeah, I can hear. Yeah, thank you, Professor. We had a nice time and we had a very good presentation with a very informative one. I hope everybody who participated here gathered very good knowledge. And please accept our small token of appreciation on behalf of Alumni Association of Peradeniya University Colombo chapter. Thank you very much for presenting this for us. Thank you again.
Thanks very much for our Alumni Association for giving me this opportunity and the honor. And I'm really pleased, and I hope I could share some of my knowledge with all of you. And most of all, Dr. Engineer Jayampathy, this gave me a great opportunity to meet our good old friends after 30-40 years, even batchmates. With our busy lives, of course, I mean, we hardly have time, and COVID still further does. So therefore, at least virtually, I'm so happy that I met so many of our good old friends. Actually, the memories refreshes. So that is the added advantage of this presentation. So I'm one of the alumni of University of Peradeniya, like all of us. So therefore, at any time, if we could do anything, not only these ones, so please tell me. from the university end. If you have anything that we could do, we are very happy to do so whatever you want to do from this university. We hope this pandemic situation will end soon so that we can actually physically gather in our old university. So we are all very welcome to University of Peradeniya. Of course, I mean, it's your property. You are the alumni, so yeah, it's more open than usual. So therefore, please come to our university and give me a call. So we will have a good time in the university as we used to have it those days. Thank you very much indeed. Thank you for listening. And I'm sorry if I have taken longer time.
Thank you,
Rajapaksa.
Thank you, Gamini.
Thanks.
Yeah. So the final item is a vote of thanks. I would like to invite our secretary, Mr. Priyat Cabral, to deliver the vote of thanks. Yeah, Mr. Cabral, over to you.
Yeah, thank you, Premalal, and good evening, everyone. Yes, so at the end of another very successful webinar, it's my privilege to deliver the vote of thanks. So, first of all, I would like to thank our guest lecturer, Professor Gamini Rajapaksa, for joining and enlightening us with your knowledge, specifically in the field of nanotechnology. Actually, you all agree if I say today's lecture was full of knowledge and interesting things on the subject matter. It gave deep insights into the topic and also revealed some interesting facts and also the points presented by Professor Rajapaksa. So once again, I would like to thank Professor for taking out time of your busy schedule and enlightening us with the knowledge, with your knowledge. Thank you so much. I would also like to thank Engineer Vipula Jayampati, the president of our association, Colombo chapter, for encouraging us to do this type of lectures or the seminars for the benefit of the society. And there will be more and more sessions like this in future. This session was coordinated by our forum committee of our association, and it is my duty to thank Mr. Premalal, who is the chairperson of this subcommittee and also one of the vice presidents. And also a big thank you to the members of the forum committee, Deepthi Bandara, Mr. Hasanta Palyakkar, Atula Kuruppu, Harshana Chaturanga. So finally, I am very happy to note almost about more than 100 participants joined this session today. So on behalf of the association, the management council and also the forum committee, I thank all the participants who joined today and for paying your attention. Thank you and wishing you good health.
Thank you very much.
Yep.
Good health to all of you. Yeah. Have a good evening.
Thank you.
Okay, so now I will end the session now. Right?
Okay.
Good night,
Okay,
everyone.
thank you.
Okay, good night. Good night.
Good night. Bye.
Thank you very
Thank
much,
you.
Professor.
Good night. Good night, everybody.
Night.
Good night, Mr. Premalal.
Don't call me sir.